I know this atoms and molecules stuff can be a little tricky, but, and you wind up with this situation. Mr. Osborne, may I be excused? My brain is full. But if we're going to talk about vaccines, we're going to talk about a tremendous number of different diseases, then we've got to talk about the building blocks a little bit, because diseases kind of funnel down to understanding a little bit about how the building blocks work, having some concept of what a molecule is, what an ion is, what the different classes of molecules, which often wind up being talked about nutritionally, are. And the next group, the molecules are getting a lot bigger now, are proteins. At its simplest, a protein is built amino acid by amino acid. You've heard of amino acids. Is that there's only about 21 that you ever find in people, and we string them together using DNA code to produce the string. And so it comes out of the little machine that makes this as this long string. Doesn't stay a long string though. Is the string cross connects and curls and bunches up, and you wind up with a molecule that has an outside to it, like a piece of Play-Doh. And you can make virtually any kind of form with a protein by winding up these strings of amino acids. And if you can make any form, you can almost make it do anything. And so proteins are incredibly versatile in terms of what it can do. So we're gonna have even if I'm gonna give you a partial list, it's a pretty decent list of the things that proteins do inside us. When we talk about mutations, when we talk about how genetics works a little bit, is that most mutations are they're in the code. Most of them are in parts of the code that don't even do anything. So a lot of mutations are kind of don't do anything. When you have mutations, little mutations in the code, the question is, does it have an effect on this? A tremendous number of mutations don't, because it's one change maybe in one, and maybe not even that. But you have a really long string wound up into this three-dimensional object. There are some where you change one amino acid on the string and the whole thing changes shape. But for the most part, changes inside this long string gives you a protein does the same thing that it would have done anyway. Is that give you an example, is that years ago, if you were diabetic and you needed insulin, you would get insulin that had been extracted from cows or extracted from pigs, because even though it had a different string of amino acids, a little bit, the parts of it that signal the liver cells, the parts of it that signal the muscle cells, the parts of it that signal the fat cells, was shaped pretty much the same way, did the same thing. So you could use it for the same function. It did turn out that there were parts of it that were not signaling parts that changed shape compared to ours. And some people actually, their immune system would go, well, that isn't right. And they would develop allergies to it because their immune system was sensitive enough to see that little bit of other difference on the three-dimensional shape and go, well, that's not our insulin, and produce a reaction to it. Reaction we'll talk about later. But the insulin functional part still works. And so you can have lots of different variants in terms of the codes, and in some cases the amino acid sequences in proteins. And as long as you don't muck around with the important part of the protein doing its job, then you're fine. As I said, this is going to be a much longer list. When we do the microscope lab and you look at your cheek cells, whenever you're looking at stuff under the microscope, I have you draw me pictures because otherwise I don't know what you were looking at. And I know what a cheek cell looks like because they have a certain shape and they have a certain proportionality in terms of how the cell relates to the nucleus in the cell. That shape is held together by strings of proteins that basically three-dimensionally are like cables. And they hold the pieces of the cell membrane together in a particular flat configuration. They hold the nucleus in a particular spot inside the cell. And you have tall skinny cells and flat cells and square cells and mobile cells. Cells are structured is changeable. All of that has to do with proteins inside that are producing the structure. As far as us, some of our structure kind of comes from the fact we have muscles under our skin. That isn't really there for structure. Your bones have some protein in them to give them a little bit of flexibility. They would break really easily without that. But your bones are actually minerals, right? Calcium carbonate. But your cartilage, flexible, stiff, that structure is almost all pure protein. Your ligaments and your tendons that hold your joints together, connect your muscles to your bones, almost pure protein. So it's an important part of our overall structure as well. True in most animals, right? Plants don't use proteins for structure. They use carbohydrates for structure. We use proteins. Some of our cells have changeable shapes. Your white blood cells can literally crawl around and do. There are filaments that pull and tug and little tubules that pull and tug and move stuff around inside the cell, move the cells, move cilia and flagella on the outside of cells. All of that movement is connected to protein systems where the proteins interact with ways that produce movement. And that's true almost across the board for all living things. Even plants that move very slowly, is that the act of a plant moving, if you've got a house plant next to a window, it orients so that the leaves are facing that light, is that what makes that movement happen, slow as it is proteins affecting the growth of the plant. Even a Venus flytrap, is that proteins are released inside the leaf. You have to send out a trigger protein and then a follow-up protein, so you have to hit the triggers twice. But what happens, this is really neat, is that when there's a fly in the Venus flytrap and it hits that second trigger, proteins flood out and make the outside of the leaves, the two leaves, grow really fast, faster than the inside, and so that it makes the thing bigger, but it also folds it around, and they have little spines that intertwine, and it captures the flies inside. Happens fairly smoothly, usually not quickly enough that it spooks the insect that it's closing around, but now it's trapped in there, and it uses other proteins to break them down and get the nutrients from them. These are plants that live in very soil that doesn't have some of the nutrients that they need, so they get them by breaking down trapped animals. Venus flytraps break down those animals with digestive enzymes. We're a big bag of chemicals doing thousands and thousands of different chemical reactions, most of which don't easily happen. They need a little bit of a kick to do what needs to be done. Enzymes are what are called catalysts. They speed reactions up, And if we didn't have enzymes doing stuff, we would be dead. Is that most of the stuff that our cells do is tied completely to an enzyme getting it to happen. In the case of digestive enzymes, they lock onto a big molecule and stress it in a way to get the pieces to fall apart. Inside your cells, if you're building, say, lipids, is that they take the bits and pieces into little slots and put them together the right way so that they build a chain that's going to eventually be the lipids, or build proteins. As the code goes through and the amino acids come in and lock onto the code one by one, is that you have enzymes there that help to build the protein. Is that your enzymes don't do everything completely, but they make the stuff that does stuff. They make the bigger carbs, they make the lipids, they make the nucleic acids, which is the last group of molecules we're going to talk about. So super, super, super important. And they have these dents that fit onto the other chemicals to make them work the way that they're supposed to. How does the liver cell know that there's too much sugar coming in through your digestive system's blood? Well, insulin goes to the liver cells and attaches to a molecule on the liver cells that's shaped like part of the insulin molecule. So you have a receptor on the cell, and then the insulin molecule fits into it. And it's a very much, a very specific kind of fit together. And there's a term I'll put after the end of the list, but I'm going to mention it now, called specificity, which is basically somebody went, well, how specific are these fit-togethers that enzymes do, that receptors do, that carriers do, that other stuff on the list does? And they went, well, sometimes it's a really specific tight fit, and sometimes it's a not so specific tight fit. So they went, well, let's take the word specific and fiddle with it. And okay, I'll give you an example of a receptor with very tight specificity. Cane sugar is, I mentioned, glucose and fructose bonded together, has a very particular shape. In your digestive system, you have an enzyme called sucrase. Cane sugar is sucrose. There's a dent that sucrose fits in, and when it fits into the enzyme changes shape slightly, stresses the bond, gets it to break. Fructose comes out, doesn't fit in the dent anymore. Sucrose comes out. Once they're split apart, they're not attached, they come off, and then it can grab more sucrose and break it down. Splenda is a sucrose molecule with 3 little changes to the outside of the molecule. 3 spots where there's a hydrogen in sucrose, there's actually chlorines on Splenda. Now, your sweet receptors have a low specificity. They'll react to a whole bunch of things and tell you it's sweet, because there's a lot of different types of sugars in the world. But it'll also react to things like NutraSweet, which is actually 2 amino acids stuck together that fit into your sweet receptors, low specificity. The sucrase, though, is so specific that Splenda won't fit into it. So you can't pop it apart and absorb it. So it goes, you taste it as sweet, but it goes right on through you because you can't break it down and absorb it, because the enzyme won't grab it and fiddle with it. So and of course, if you're going to change shapes of things with mutations, how tight or loose these things are is very tricky. Right now the question is, one of the mutations on coronavirus affects the molecule that attaches to a molecule on your cells to get into it. And now they're going, does it make it a tighter fit or looser fit? And you may go, well, a tighter fit would be better, right? Maybe not. Maybe a tighter fit wouldn't help it get in. Maybe it needs to have a certain level of looseness in order to interact right with the membrane to get the viruses into the cell. You never know with this stuff. Your cell membranes are pretty good barriers. You need to move stuff in and out of the cell that won't actually go through the membrane on its own, because it's dissolved in water. Water doesn't go through very easily, or the molecules are too big, or the atoms are charged, or whatever it is. And So in your cell membranes, there are molecules that will help stuff go in or help stuff go out, whichever way it's supposed to be going. Those are carriers. They do just exactly what this sounds like they're doing. Also, there are carriers that can help you move stuff around in your blood, things that don't dissolve terribly well. Certain minerals don't dissolve real well. Lipid parts, like cholesterol, terrible at dissolving in water. They need to be attached to a protein carrier, and even that doesn't dissolve that great. Those are called lipoproteins. We have low-density ones and high-density ones. For those of you that had blood tests, you did your cholesterol test, and then you have your LDLs and your HDLs. LDLs, low-density lipoproteins. HDLs, high-density lipoproteins. They're like grains of sand in a stream. As long as the stream is moving okay, they're moving with it. But if the passage of liquid slows down too much, they can settle out and they're a little bit sticky. They'll stick to the lining. And that's what happens over your lifetime, is that the LDLs going around curves in your arteries where the flow is fairly quickly will settle out like a sandbar on a stream and build up, and then you'll clot over the bump. And then it'll build up on that and it'll clot over. And it can actually eventually close your whole artery down over time. That's what, atherosclerosis is, a buildup of stuff in your blood. They have to go through like either a little squeezy thing that smooshes the lipids flatter against the lining of the artery, or a little rotor-rooter thing that kind of grinds it off. But it's the lipoproteins that are, your lipids wouldn't even go through your blood if it weren't for the lipoproteins doing the carrying. So there's a lot of different types of carriers. I'm still going down the list, but I'm running out of room here. So right now, you have different types of white blood cells that are basically crawling through all of your tissues and on your surfaces, and they are coming up against big molecules and they're kind of tasting them. And depending on how they fit, specificity, they know what's supposed to be part of you and what's not supposed to be part of you. They're your bouncers, your gatekeepers for your immune system. If something isn't right, they will react, is that you'll get a kind of a general response. We'll talk about this when we get to the immune system. And so you have molecules that will slow viruses down or possibly affect cells that have viruses in them. And so when you get sick with something, that's pretty much what's going to make you get over it. Because this part, the antibodies that you're going to make, are fairly slow to get made. But what does happen is some of your white blood cells will take this molecule off of a virus invader or a bacterium, or in some cases like a toxin, from a bee sting. And they'll pop it off of where it came from and give it to another cell, orient it the right way, and say to the cell, okay, this is an invader. We don't know if it's bad or good, it just doesn't belong here. Do something about it. And what will happen is your other cells will make a special type of protein called an antibody, where it's got this bottom, they always have, and then it makes a dent that will specifically fit on that molecule it was given. This is called an antigen. It generates an anti-response. And this is an antibody. You actually have two spots that attach. So, okay, so I had COVID in March. Is that my cells popped molecules off the surface of the virus, fed them to my beta cells. My beta cells have made antibodies. I had the antibody test. And so I now have these molecules in my circulation that if I get exposed to coronavirus, they will immediately attach to this molecule on the coronavirus. So this is the attachment end to the antigens. But it changes the shape of the protein. When it changes the shape, this end becomes a signaling end. It says to immune system cells, it's like, hey, come do something about this. And the immune system cells come in and they go to the other end of the antibody and they just, there's a bunch of different things they do. They poison it, they break it apart, they eat it, Basically, they're going to wipe out these invaders before they can set up shop in you. That gives you the immunity to not getting infected again. Right now, they're talking about your antibody levels drop, but the important part of your immunity is those cells that you built in the first place are like your immune memory. And as long as they're capable of cranking antibody outs at a moment's notice, you've got the programming in for these antibodies, you're still going to be immune down the road. That's apparently what's going on with COVID. But a vaccine exposes you to the antigen without giving you the virus that's going to make you sick. So you have antibodies that will attach to the virus and keep you from getting sick without you having to get sick in the first place. And that's how a vaccine works. Sometimes they have little chemicals to kind of exaggerate the response, because the response, you can't always trust the level of response, but they're not dangerous for the most part. So, we have antibodies. They attach to antigens, which are usually big molecules. This is also a function of allergies, and there's some things that people are allergic to that they're really chemically sensitive to. There is a difference between a chemical sensitivity like you might have to certain metals. Some people go, I can't wear rings with that stuff in it, it makes my hand turn green. And they go, I'm allergic to that. Well, you're not. You're sensitive to it. It's still a reaction. Probably doesn't matter to you what we call it. But it's not technically an allergy. An allergy is an antibody response, and then very often an inflammation response to what your immune system decided was an invader. Keep in mind, your immune system doesn't wait to find out if these antigens are dangerous or on something that's harmful. They just go, this is foreign, we need to make antibodies to it. We'll talk about it later when we talk about the immune system. Okay, so that's the list of uses. There's, I want to get this written down, I've talked about it already. Specificity is an important feature because proteins so often fit on other things, and so how specific they are for the lock can be very, very important. Your taste receptors tend to be very broad. They taste a lot of different types of molecules and register as sweet or sour or bitter or whatever it is. And your enzymes tend to be pretty specific. Your antibodies, you hope, are specific. If your antibodies are less specific, then they sometimes will make the mistake of being made for an invader, but it will actually fit on a molecule on your own cells. This is what they call autoimmune diseases. For instance, if people with type 1 diabetes have antibodies that fit onto their insulin-making cells in their pancreas. They fit onto a particular molecule that's on those cells. Why did you make those antibodies? The assumption is you were exposed to some sort of virus. In fact, they've got a pretty good idea what virus it is, because they're finding that people vaccinated or kids vaccinated for that virus are much less likely to develop type 1 diabetes. Okay, I go off on tangents. But you make an antibody that will fit onto the invader, but it also flags your own cells. And your immune system doesn't know. If the flag goes up, man, they come in and kill it. So you may have rheumatoid arthritis where it goes after collagen molecules in your joints, or Addison's disease where it goes after some of the cells in your adrenal glands, or multiple sclerosis where it goes after the insulators on your nerves. But what's happened is you made an antibody to something legitimately foreign that isn't quite specific enough that it also makes the mistake of attaching to molecules on your own cells. So specificity is a big, big player in a lot of diseases. Speaking of diseases, and these will come up later, but I want to talk about it here. There's a type of protein called a prion. These are bizarre. The closest thing that you can find in nature to honest-to-goodness zombies. A prion, the string of amino acids, matches the amino acid string in one of your molecules that's doing something in your body. In the case of Creutzfeldt-Jakob syndrome, it's a molecule that's active in your nerve cell, in your central nervous system, in your brain. You get the prions, mad cow disease is an example of this. The prions get into your cells, and this is what they can do. They attach to the molecules in your cells that match their string, but they're a different shape. When they let go of your molecules, it's now the shape the prion was. So it's no longer doing the job it was supposed to do. It's now capable of grabbing more of your molecules and turning them into prions. Remember I said they're like zombies? So they come in, they grab a functional molecule that they match the string of, but not the shape of, and they change its shape into a prion. That goes on to do it to another one, to another one, to another one. And Creutzfeldt-Jakob, sometimes it's a prion that works really quickly. Mad cow disease can affect you in a couple of weeks. The classic Creutzfeldt-Jakob syndrome can take 20 or 30 years before it has a big enough effect that, because the more molecules it changes shape of, the fewer molecules are doing whatever the heck job that molecule had, and that's going to affect the function of the overall cell. Okay, that's it for proteins. That was a pretty big section.